everybody. I'd like to welcome you to today's Congressional Biomedical Research Caucus presentation. First off, I'd like to thank the co-chairs of the caucus, and that is Representative Brian Bilbray from California, Representative Charlie Dent from Pennsylvania, Representative Jackie Spear from California, and Representative Rush Holt from New Jersey for their commitment, dedication, and ongoing support of the caucus. I'd also like to thank the Howard Hughes Medical Institute, who supports the caucus through a generous grant. We are very grateful for their continued support. As you can see, we do videotape every briefing, and you can find past briefings on the CLS website at coalitionforlifesciences.org. Now I would like to introduce our speaker today, Dr. Eric Olson. Dr. Olson is a professor and chairman of the Department of Molecular Biology at the University of Texas Southwestern Medical Center in Dallas. At UT Southwestern in Dallas, Dr. Olson holds a Robert Welch Distinguished Chair in Science, the Annie and Willie Nelson Professorship in Stem Cell Research, and the Pogue Distinguished Chair in Research on Cardiac Birth Defects. Prior to his appointment in Dallas, Dr. Olson was both a professor of biochemistry and molecular biology, as well as chemistry at the MD Anderson Cancer Center in Houston. He holds a BA, PhD, and honorary doctorate degree from Wake Forest University in Winston-Salem, North Carolina. As one of the world's leading experts in developmental biology of the heart, Dr. Olson has received numerous domestic and international awards from organizations such as the American Heart Association, the National Institutes of Health, and Columbia McGill Universities, among many others. He is a fellow of the American Academy of Arts and Sciences, U.S. National Academies of Sciences, and the Institute of Medicine of the National Academies of Sciences. So without further ado, Dr. Olson. Thanks.
Thank you, Lynn, for the introduction, and thank you also for the invitation to be here. I'm really happy to be here today. It feels just like Dallas outside. So my lab studies the heart, and this is a diagram of the heart. The heart is the most important and perhaps one of the least understood tissues in the body. Keep in mind that the heart beats one time per second from the beginning of life until the end of life, sometimes 80 plus years. It never skips a beat, and the first time it does skip a beat is essentially the last time it beats. The heart has really been a focus of intense fascination by philosophers, physicians, artists for centuries, and really has remained highly mysterious. And what I'd like to do today is to talk to you about where we are with our understanding of heart disease and what some really new out-of-the-box strategies are to begin to repair the heart. This is the first painting of the heart. It's actually graffiti from 10,000 years BC in the Pindal Cave in South Spain. You can see here there's it's a woolly mammoth, and that's the first depiction of the heart. So graffiti was probably more meaningful back in those days. This is the first three-dimensional rendering of the heart that's ever been discovered. It's the Olmec effigy from Mexico, dating about 1,000 years BC, and you can see that even at that early time, it was known that they had it pretty good. They had the two ventricular chambers. This would be the right ventricle and the left ventricle of the heart. They had these two structures, which are the atrial chambers of the heart, and they even had the blood vessels entering and leaving the heart appropriately. I'm not sure what this structure is, but I think that was maybe lost in evolution later on. The most famous physician of all time was William Harvey, almost 500 to 600 years ago. He was the first individual to recognize that there are two chambers of the heart, the left and right ventricles, that pump blood through separate circulations to different parts of the body, and he described that in the most famous medical textbook of all time. Philosophers have also wondered about how the heart works and how it does what it does. Even contemporary philosophers have questioned how or why their heart does what it does. So, given the fact that the heart has been a central focus of so much attention for thousands of years. It's really remarkable and humbling to realize that diseases of the heart are the number one cause of morbidity and mortality in the world today. As I think many of you know, myocardial infarction, which occurs when the blood flow to a portion of the heart is blocked through a cholesterol blockage. This leads to death of heart muscle cells, as shown here, and a loss in the ability of the heart to pump blood to meet the demands of the body. If you keep in mind that a myocardial infarction takes place every 25 seconds in the United States, and up to half of these heart attacks are ultimately fatal. It's really humbling to realize the magnitude of this disease and our, so far, relative ineffectiveness to treat it. A little bit of anatomy here for those of you who aren't familiar with the heart. This is a schematic rendering of the heart, looking at it from the outside, and this would be looking at it from the inside. The heart has two chambers that pump blood to different parts of the body. The left ventricle, shown here, pumps oxygenated blood out to the rest of the body, and then the right ventricle receives the deoxygenated blood back from the body, and then it pumps that blood out to the lungs in a closed circulatory system that I think many of you are familiar with. The heart is a remarkably dynamic organ that can adjust its size and its function and its contractility to meet the demands of the body. So under normal conditions, for example, during pregnancy or in response to extensive exercise, the heart increases in mass and it enhances its cardiac output to pump more blood to provide oxygen to the tissues. But in response to a number of disorders, the heart undergoes pathological changes, and some of the stimuli that can cause the heart to undergo these pathological changes would be high blood pressure or a heart attack, as I've described to you, or chemotherapy. Many people are unaware that heart toxicity is the major dose-limiting toxicity in patients on chemotherapy, and this is a particularly severe situation in women being treated for breast cancer because the heart is highly sensitive to breast cancer chemotherapy. These types of insults lead to a series of changes in the structure and function of the heart that include the death of heart muscle cells, fibrosis, which causes stiffening of the heart, loss of pump function, cardiac arrhythmias, where the heart develops an abnormal electrical beat, and then ultimately these changes can culminate in heart failure or sudden death. To put this in a human context, heart failure, that is the inability of the heart to pump the necessary amount of blood needed by the body, is the number one cause of death and disability in the United States. The number of patients with failing hearts exceeds the number of patients with all cancers combined. It's estimated that there are 5 million Americans right now suffering with heart failure. There is no cure. If one is diagnosed with heart failure by your cardiologist, you have a 50% chance that you will not be alive in 5 years. And the consequences on the healthcare system are really staggering. To put that in a global context, this slide illustrates the major causes of death and loss of years of life worldwide. And you can see that ischemic heart disease, again resulting from blockade of the coronary arteries and a myocardial infarction, is the number one cause of death and disability in the world. So obviously this is a very important problem that scientists and physicians have been focusing on for many years, and while there have been some advances, it's also clear from the numbers that the advances are inadequate, and there is a huge need for further research and further understanding of the basic mechanisms of how the heart functions, how it dysfunctions, and how one might intervene in heart disease therapeutically. So what I'm going to do today is to talk to you about some work that's come from my lab in which we're trying to develop really new approaches to push on the envelope of ideas of how we might restore the function of the heart. This slide illustrates some of the problem that I'll be talking to you about. This is a cross-section, a histological section through the heart of a mouse, but it could also be the heart of a human. They look just the same. This chamber here is the left ventricle of the heart, and this small chamber is the right ventricle of the heart. When an individual has a heart attack, the blood flow to this portion of the left ventricle is blocked, and so all these heart muscle cells die because they no longer receive oxygen. And what happens is that these muscle cells are replaced by a scar, which you can see there stained in blue, and this causes dilation of the left ventricular chamber, and the ability of the left ventricle then to contract and squeeze blood is compromised, and this causes heart failure. The human left ventricle has about 2 to 4 billion heart muscle cells in this left ventricular chamber that are responsible for pumping blood to the body. And even a transient, what would be called a transient ischemic attack, where there's just a temporary block of blood to this region of the heart, can kill 25% of those heart muscle cells in just a few hours. And the problem is that the adult human heart cannot generate new heart muscle cells. And so there's no way for the heart to repair itself. The heart is distinct from other tissues. For example, as you know, if you cut your skin, the skin can repair itself completely seamlessly. The liver is also a fascinating tissue. If one surgically removes 50% of the liver, the rest of the liver will grow back a complete new liver, exactly the same size and structure as the original liver. But for some reason, that mechanism has been lost in the hearts of mammals, including humans. And so what I want to talk about today are some strategies one might use to reawaken or to activate this regenerative program in the setting of heart disease. Now, many other organisms are able to regenerate their hearts. In fact, they can regenerate all sorts of structures. Various fishes and salamanders or newts have remarkable regenerative powers, as I think many of you know. If you amputate, for example, the fins, the legs, or the tail of a salamander or a fish, they will grow back the limb to exactly the same size and structure and function as the original limb. How is it that these animals have this program built into them where they can simply regenerate a limb? And the same goes with the heart. If you surgically amputate half of the heart of a newt or a fish, they will grow the heart right back to the exact same size and structure. So we know, in principle, there must be biological pathways and genes and mechanisms that can do this. The question is, why are they switched off in the adult heart of humans, and can we activate these pathways? We know that the adult human heart, as well as the adult hearts of other mammals like mice, cannot regenerate. But one question our lab had was, is it possible that early in life these regenerative processes exist and maybe they're just shut off as animals age? And this was a simple experiment that some of my students did, which I thought would never work, and that's probably why it did work. So they took baby mice and they surgically amputated the apex of the heart, which you can see schematically here. They removed 20% of the apex of the heart, and then they asked, is it possible that a newborn animal might still retain some ability to regenerate that's lost later in life? And remarkably, they found that these hearts could completely regenerate back to normal, and you can see the time course of that here. So here again, this is a cross section. This is the left ventricle. They sever the apex of the heart, and on day 1 they see a clot forms there to plug the leak in the ventricle. On day 2 they see inflammation of this region. By day 7 they see muscle cell proliferation, and by day 21 there's a complete disappearance of the wound. The heart is completely restored to normal structure and function. This is a simple but I think elegant and important experiment because it says that the mechanisms, the genes, the proteins, the signals that are required to regenerate the heart really do exist in a mammal, such as a mouse and presumably in a human. But somehow these are silenced later in life. And so if we could find the biochemical key to unlock that process, it could have enormous implications. my students wanted to understand, when does the heart lose this ability to regenerate itself? And so they did what would be called a time course experiment. So here, this is a cross-section of the heart cutting through the heart this way. So this would be the left ventricle and the right ventricle. If they injure the heart on day 1 after birth in a mouse and they look 21 days later, the heart is completely regenerated, absolutely indistinguishable from normal. If they delay this a bit and they injure the heart at 1 week of life and then they let these animals go for 21 days, you can see the regenerative process is starting to diminish. Now there's a scar formation here, and if they wait till day 14, the scar is even more pronounced than on day 7. And so from these and other experiments, we know that there's an inverse relationship between the ability of the heart to regenerate and its ability to form a scar. So as scar formation, so-called fibrosis, increases, regenerative properties decrease. So the scar is essentially a blockade. It's like putting a big barrier in the road to regeneration, and it prevents these hearts from repairing. So one would like to then try to develop strategies that could diminish fibrosis with the hope of enhancing regeneration. So this is simply a cartoon to tell you what I've shown you so far, and that is the early heart in the mouse, and we believe this also applies in the human, can regenerate. And the regeneration process follows a specific series of biological events that ultimately culminates with the disappearance of the wound and the restoration of function. And so the question is, can we unlock this lost mechanism in adult hearts of mice or humans? Clearly, heart failure therapy in 2011, despite decades of effort and billions of dollars in research, still has a long way to go to solve this major health epidemic. Right now, there are essentially 3 major approaches that are taken in the clinic to treat individuals with heart failure following a myocardial infarction. The most common would be the use of drugs such as beta blockers or various calcium channel blockers, but clearly these are inadequate to restore cardiac function, or heart failure wouldn't be an epidemic that's affecting millions of people with no cure. Heart transplant is another alternative approach one could take to restore cardiac function, but obviously this is not a practical approach for reaching millions of individuals, particularly those in third world countries. And other devices such as LVADs, which you've recently seen in the news, Dick Cheney has one of these, and pacemakers and other such things, can also be used to temporarily enhance cardiac function in the setting of heart failure. But again, these are expensive, they're cumbersome, and they're highly limited with respect to the number of patients that can receive these sorts of technologies. An alternative approach is to use biology and let biology guide you to try to understand the basic mechanisms of heart repair and see if we could exploit those therapeutically. And what I'm going to talk about for the remainder of the talk are 2 approaches. One are to use stem cells, which you've all heard about, to inject them into the heart. And another, which we favor, is to use the intrinsic ability of the heart to regenerate, which we know exists, and just to try to increase the efficiency of the heart to do that. There are many types of stem cells that you've heard about, and there have been literally dozens of human trials now that have been going on for over a decade, where various types of stem cells have been injected into human hearts to try to improve cardiac function following a myocardial infarction. Stem cells have been generated from blood, bone marrow, skeletal muscle, fat, and even embryonic stem cells. Bottom line is there's been minimal benefit reported in patients following long-term treatment with exogenous stem cells. And the reasons are there are many challenges that are facing stem cell research. Many of these will ultimately be dealt with appropriately, but they remain significant challenges to keep in mind, particularly with respect to treatment of the heart with stem cells. Regarding the use of human embryonic stem cells, I think many of the difficulties with this approach are obvious. They're very inefficient in giving rise to cardiac muscle cells. They can do so, but they do so at a frequency of less than 1%, so this is not an efficient approach. There's obvious ethical issues with respect to obtaining embryonic stem cells. There's immunological rejection. If you take cells derived from ES cells from one person and put them into another, the immune system will reject them. And embryonic stem cells are highly tumorigenic. And so if one injects cells derived from ES cells into a person, you have to be absolutely certain there's not even a single contaminating ES cell, undifferentiated ES cell in there, it will cause a tumor. Now, there's a recent approach that's been in all the news, and I think all of you have likely heard of it. It's called induced pluripotent stem cells, or iPS cells. And this is a fascinating technique where you can take the skin cell from an animal or a patient, and you can put 4 genes listed here into that skin cell, and it will turn that cell into what's called an induced pluripotent cell, which is essentially like a stem cell, and it can form heart muscle cells with low frequency. Problem is, iPS cells also are highly effective at inducing tumors. This process is slow, and recent papers just came out in the last month that said that cells derived from iPS cells are immunologically rejected in patients. So you can imagine the FDA is highly unlikely to approve therapeutic approaches that have high likelihood for tumor formation, as with these various cell types. And finally, there are many types of what are known as adult stem cells that you're all familiar with. These come from various adult tissues, in particular the bone marrow and other cell types. But the problem with these is the effects are modest, and there's no proof so far that these can give rise to heart muscle cells in an animal. In fact, when one injects these, they disappear almost instantly. So there's many major challenges with respect to this approach. So in summary then, the common obstacles for using exogenous stem cells to inject cells right into the heart are many. They include that this is a technically cumbersome and expensive approach. I think it's very difficult to imagine that one would be able to efficiently harvest stem cells from millions of patients worldwide who suffer from heart failure, and then to go through all the technology to bring them into the clinic, open up their hearts, inject them into the hearts. Although this will work for some patients ultimately, and maybe it will be optimized, there's still many challenges. There's poor survival, and also the heart, as you know, is highly electrically integrated. And if one disrupts this electrical integration by injecting foreign cells into the heart, it can cause arrhythmias, and arrhythmias are the major cause of cardiac sudden death, where the heart just suddenly stops. So we're experimenting with an alternative approach, and that is to try to harness the intrinsic ability of the heart to regenerate by introducing either genes or molecules into the heart and see if we can stimulate this regenerative process. So the heart contains a stem cell population. It's called the epicardium. It surrounds the heart. You can see it here in green. And these cells serve as a reservoir of possible stem cells for the heart, but clearly their ability to rejuvenate the heart is inadequate under normal conditions, or heart regeneration would be commonplace. This is a cross-section through a human heart, actually, and here you can see the epicardial layer surrounding the entire heart. And this is the heart of a mouse by comparison. So this epicardial layer that surrounds the heart serves as a source of progenitor cells or stem cells. And we know from developmental biology studies that these cells can form many of the different tissues and cell types of the heart, but it's a relatively inefficient process, and so we would like to enhance that process using various drugs. In this experiment, we labeled some of those epicardial stem cells with a marker that turns the cells green, and here you can see, looking at these hearts, green fluorescent heart muscle cells, confirming that, in fact, these cells can give rise to heart muscle cells, but the process is inefficient. So the approach we've taken is to ask, can we discover new drugs that could stimulate these heart stem cells to efficiently give rise to heart muscle and to repair the injured heart following a myocardial infarction? And so to do that, we've taken stem cells and we've treated them with 250,000 different drug-like molecules that are in what's called a drug library, and we've asked, are there any of those that can make heart muscle cells from a stem cell? And the way you do that's with a robot. So here's a robot. These are wells of cells with 384 cells in each well, and this robot delivers a different drug to each well, and then we screen, as I said, 250,000 different compounds. And remarkably, we identified a number of fascinating chemicals that were able to turn on heart muscle genes and to push stem cells in the direction of the formation of heart muscle cells. And without going into the details, these are the structures of many of these genes, and we call this our top 10 hit list. This is really a treasure chest now of biologically active small molecules that we can test in animal models for their abilities to promote heart regeneration. And here you can see we've taken some cells in culture, and here are some stem cells that have been converted into heart muscle cells. So you can see they're beating rhythmically in a tissue culture dish. And on the bottom would be just looking at the cells on a dish, and if we treat with the drug, we can turn on this heart muscle protein which stains these cells in red. So we're keenly interested in how these drugs can be used in vivo or in a whole animal to restore cardiac function following a myocardial infarction, and we're currently doing animal studies to test that. The second approach would be that we're taking is to ask, could we identify any genes that direct the formation of heart muscle cells, normally in an embryo or in a fetus, and if we could find those, could we reintroduce those into the scar of an infarcted heart, and maybe that could rejuvenate the heart and regenerate heart muscle cells. So this is a bit of a complicated experiment, but here's the way that this experiment is done. If one takes a mouse and ties off the coronary artery right here, the rest of the heart will die. It has a severe heart attack. And when one does that, there's the heart muscle cells shown here in rectangles all die, as you know, and that gives rise to a scar. And there's another cell type in the heart called a fibroblast. This is actually the major cell type in the heart. And they get amplified, and they give rise to the formation of the scar. And so what we've asked is, can we discover any genes which, when injected into these fibroblasts, would turn them into heart muscle cells and would restore cardiac function and at the same time prevent fibrosis, which, as I told you before, is a major barrier to regeneration? And the answer is yes, and this is one experiment. So here's an experiment with a mouse where we cause the mouse to have a heart attack by tying off the coronary artery, and then this entire region of the heart dies and is left with a scar, which compromises cardiac function. You can see the scar stained there in blue. And here we've injected this heart with 4 genes, which we knew from studies in developmental biology were necessary and sufficient to generate heart muscle cells in an embryo. And when we inject those 4 genes, we can rescue these hearts and severely diminish scar formation and improve the formation of new muscle cells. So now we're testing all sorts of different genes, and we're combining them with the drug molecules that I described earlier and asking, are there any interesting combinations of these genes and drugs that could regenerate the heart? And we're asking what cells in the heart might be most susceptible to becoming heart muscle cells in response to these stimuli. Now, the heart, remarkably, is surrounded, particularly in humans, is surrounded by a layer of fat. You can see that here schematically, and this is a human heart. Sorry, it's a little dim, but you can see the human heart is also covered with a layer of fat. And so we've wondered, might it be possible to take this reservoir of fat cells that just engulfs the heart and find a combination of genes or drugs that could turn those cells into beating heart muscle cells following a heart attack as a way of restoring cardiac function? And so one of my students did that experiment, and he brought me this movie. I don't know if you can see this. This was a plate of cells. These are fat cells that he isolated from the region surrounding the heart. And he put those same 4 genes that we knew could generate heart muscle cells when we injected them into an infarcted heart. He put them onto these cells, and they start spontaneously beating. So you can see it. And fat cells don't normally spontaneously beat. They often jiggle, but they don't beat. So we are really interested in this possibility that one can take these combinations of genes and drugs and exploit this population of cells that exists normally in the heart, namely these fat cells or maybe fibroblasts or others, and turn them into beating cells in vivo in the native environment of the heart. And this would then eliminate the need to inject cells from another source into an injured heart. So What I've told you then today is, as our approach is to try to find new strategies to repair the heart following a myocardial infarction. And we're keenly interested in trying to exploit these new regenerative genes and new regenerative molecules to extend this work both into large animals and eventually into humans. So the work I've talked to you about was carried out at the University of Texas, Southwestern Medical Center in Dallas. Here's downtown Dallas. The medical center, my laboratory is here, and this is all the UT Southwestern Medical Center where this work was carried out. And this is the building where my laboratory is located. This work would not have been possible without a lot of support. We were fortunate. We received one of 3 National Stem Cell Research Center grants from the National Institutes of Health, the NHLBI in particular. And we've also been strongly supported by the American Heart Association for our work on cardiac stem cells. Now, I'll just mention in closing, since Lynn mentioned that I hold the Willie Nelson professorship. So Willie Nelson, the famous Texas icon, as you know, he's involved in supporting many philanthropic causes. He learned that I play in a rock band with people from my laboratory. We're called the Transactivators. And this is us playing a gig in Albuquerque. But so people always ask me, what the heck is a transactivator? So I went to the source of all knowledge, Wikipedia. Transactivation is an increased rate of gene expression triggered either by biological processes or by artificial means. And transactivation can be triggered by transactivators. So that's the basis of our band. And so Willie got all excited about this, and he raised money in a benefit concert to create the Annie and Willie Nelson professorship. And we've started a grassroots organization in Texas to support stem cell research. And in fact, we're going to generate stem cells from Willie Nelson, and we're going to call them Little Willies. Okay, so being a Willie Nelson professor means I go backstage with him often at concerts. This is Willie and me on his bus before a recent concert. I can tell you that Willie Nelson prepares differently for his public performances than most scientists, or certainly than I do. But he's really supportive of this effort. And lastly, here's one of my favorite quotes from Willie that I'm still trying to understand. He's an avid golfer. So par is whatever I say it is. I've got one hole that's a par 23, and yesterday I damn near birdied the sucker. So I don't know what that means. I think it means that if you set a goal and just keep trying, you can ultimately achieve it. Or it might mean that Willie just has trouble focusing on the hole. Anyway, that's all I have to say, and I appreciate your attention. Thank you.
I've got 2 questions. First of all, you talked about the fat cells around the heart. Are fat cells in other portions of the body different so that they are immunosuppressant? You would have to do that if you were taken from other parts of the body. And the second thing is, how politically difficult is it in a very conservative state like Texas for you to deal with stem cells?
Those are both good questions. Our work on the potential conversion of fat cells into heart muscle cells is very new. It's not yet published. I think, as you're pointing out, there are many different types of fat cells in the body. Some might be more susceptible than others to this process that I described. And that's precisely why we've taken the approach to trying to convert the resident fat cells that normally exist in the heart into cardiac muscle cells, because we feel they're in the appropriate cellular environment for that, and we think maybe they're already kind of partway down that path. But the second important reason is because it does not require cellular transplantation. Most of these stem cell studies that have been done where people are injecting cells directly into the heart or they inject them into the coronary arteries and hope they go to the heart are, as I said, extremely cumbersome, ineffective, and can cause arrhythmias. So that's why we're just trying to enable the heart to do what it used to know how to do. It knew how to regenerate earlier in life, and it's somehow forgotten, and maybe we can reawaken those memories. And so that's the approach we're taking. With respect to stem cell research in Texas, it has not been a problem whatsoever. I'm not here to get into the politics of it. Most our work has not been done on human ES cells derived from human embryos. We've focused to date on trying to figure out what are the basic principles and the basic mechanisms of stem cell biology that would enable a stem cell to form a heart or to form something else, and can we find drugs that do that? And these principles are largely conserved in different organisms. So if we can figure it out and optimize it in a mouse, then we believe that'll be applicable to humans. And lastly, though, just to come back to your point, again, I think the approach we're taking, and it's not driven by politics at all, it's driven by pragmatism. That is, the approach we're taking is not to try to take ES cells, human or otherwise, to generate heart and muscle cells and put them back in a heart, because we just don't think that that is a practical approach, and we think there are better ways to go than that.
I'm interested. I actually am a cardiovascular neurophysiologist from Ohio State, and I taught at Michigan, so I'm fascinated as to what you presented today. But I'm wondering, has there been any work on two parts, on the stellate ganglia in terms of looking at it as it changes the dynamics of the heart? Any recent research at all?
I'm not aware of any. So no, I'm not sure.
Projecting the stellate ganglia, I was reading in some of the HHS things.
As a way of generating maybe the conduction system or something. Yeah.
Very interesting. But regeneration of nerves, and the other section I was interested in is that some people grow collaterals. They have a heart attack and then they grow collaterals. Is there any key to knowing how we can take that and translate that? Because that is a way people survive. They do have a heart attack.
Yeah. So the question that's being raised is a good one, and that is oftentimes if you have a blockage of a coronary artery, the artery will sprout collateral vessels that try to feed the injured tissue. So that is one approach for restoring blood flow, but the problem with that is that it takes a while to generate these collaterals. But really, all these heart cells, the billions that I described to you that die, they're dying very fast, within minutes or hours after the infarct. So it's not going to, that approach would probably work really well, the approach you're describing, in conjunction maybe with some of these regenerative approaches that we're describing.
And thank you.
I was wondering, what is the time scale on the heart to go from the scarring to the regeneration? You never specified a time, and is it going to be, or is it currently, or will it in the future be a feasible option?
Those are good questions. So most of our work on the time course has been done in this newborn mouse model, and we believe that the regeneration takes place over a couple of weeks. So if we surgically remove the apex, 20% of the heart, within 14 to 20 days, that 20% is completely regenerated. So it's quite a very rapid process. Your second question?
The question was, how does it change with the introduction of the artificial molecules?
So that's a good question. That's exactly what we're doing right now. We know that these molecules and genes can enhance this process, and right now we're trying to figure out what is the exact proper recipe for heart repair. And as you can imagine, the exciting part is discovering the genes and finding the new drug and doing the biology. The less exciting part is kind of fine-tuning at least for me, fine-tuning all the details. And so now you have to figure out what is exactly the right dose and time for delivery and those sorts of things. But those are important, and many of those issues need to be worked out probably by biotechnology or drug companies. And in fact, there are a couple of biotechnology companies that have sprung up from this work and are pursuing those sorts of questions.
I know that NIH grants come in all sizes, and there was some discussion earlier this year about reducing the average size of an NIH grant from about $430,000 to $400,000. Do you mind giving us a sense of the size of the amount of financing that it takes to support the kind of work that you're doing? I mean, I really have no concept. I don't think you're in the $250,000 category.
It takes a lot of money to support this kind of work, and the type of work that I described to you today would be classified as discovery-based research. And sometimes the study sections at the NIH are less interested in funding discovery-based research than they are in filling in the details once you already sort of have the sketch. Discovery-based research sometimes can be called fishing expedition because you don't know what you're going to get or if you're going to get anything. Clearly, both types of research are important. It takes funds from many sources to run an operation like my laboratory, so I have benefited enormously over the past 30 years of my career from NIH support, and I continue to have NIH support, knock on wood. But it could not be done solely with just NIH support. It takes a lot of other supplemental sources of resources.
What can naturally occurring late-stage differentiation that already happens in the body teach us about the techniques you're trying to use? And primarily I'm thinking of olfactory epithelium with like basal cells differentiating into receptor cells. We understand that occurs today. Can that process help us learn about potentially turning fat tissue of the heart wall into...
Well, that's a good question. The process that you're describing is a process of developmental biology, and I'm a developmental biologist by training, so I tend to believe that most disease processes involve the same cellular circuits and genes and mechanisms that underlie development. And even the differentiation of adult tissues is basically based on the recapitulation of a developmental process. So I strongly believe that basic research into fundamental mechanisms of developmental biology is an absolute necessity for understanding disease. And I think you can't, I think it's obvious to all of you who are in this room, you can't necessarily anticipate where the discovery is going to come from. I mean, once you understand a process, such as heart development or something, unexpected linkages to disease will reveal themselves. And so while directed research to find the cure for heart failure is certainly that's important, in many cases that's not where the cures come from. Okay, thank you very much.